All right, thank you very much for the introduction. Thank you very much for the education on IT. So I'm going to be talking about recent clinical trial results, which have been testing iron chelation for the treatment of Alzheimer's disease and Parkinson's disease, and talk about the implications of those results in our understanding of the pathogenesis of these disorders. So Alzheimer's disease is well known as a disease that's got a prominent neuropathology with amyloid plaques and neurofibrillary tangles, but it's plenty of other pathologies that occur within the brain, and I'm listing them there, things like iron accumulation, oxidative stress, mitochondrial dysfunction, glial activation. And what is not known is really what is the sequence of the events that occur that induce these features. I'm going to go to the... Right. So that's a picture of the iron accumulating within the neurons in Alzheimer's disease. Next, the picture of the amyloid plaques filled with iron. Next, and there's an activated microglia which fill with iron. So we spent the better part of 10 years looking directly at the iron within the human brain, studying postmortem tissue and using imaging as well as biomarkers to see whether the iron co-pathology of Alzheimer's disease has any predictive value in terms of the outcome of the disease. And this is just a sampling of some of the data that we published. So in this study, which is a collaboration with Martha Claire Morris and Julie Schneider at Rush University Medical Center, you see on the this is the measurement of iron levels within a series of 685 brain samples which are kept at the Rush University Medical Center. These are individuals who, when they die, there has been up to 10 years of cognitive testing that has been done for each volunteer, and so it could track their change in cognition over time up to the 10 years prior to their death. And the people who died with the highest level of iron were the ones who had the most accelerated cognitive decline, which you can see on the y-axis, the x-axis being years prior to death. Next, Amit. We also studied iron burden with QSM. Go back one. Amit. Sorry. Go forward one.
Oh, I think you can try to use it now.
Yeah. Okay.
Yeah.
Oh.
Yeah.
All right.
But you prefer to say a bit.
I have to, but I think you can try to use it.
All right.
Okay.
Yeah. All right. That's better. All
Oh,
right. Thank you.
I'll stay by your side. It makes me feel good.
Okay, and these are data for using QSM, which is we're going to be talking about at this meeting a bit, I believe, which is a way of using the MRI to measure brain iron. The iron levels are on the x-axis, and this is in the hippocampus, which is severely affected in Alzheimer's disease, and on the y-axis is the change in the Z-score for cognitive testing over a period of 6 years, and shows the rate of change. These are a series of patients who have either got amyloid in their brain, which is the red line, or have no amyloid in their brain, which is the blue line. And it's the patients who've got amyloid within the brain who are sensitive to the elevation of iron. So the rate of cognitive deterioration accelerates in proportion to the amount of iron. So there's a synergy between iron and amyloid pathology in Alzheimer's disease, which seems to be associated with a far more rapid cognitive decline. Oops.
It's the opposite side.
Because it's Hebrew. And finally, using ferritin, which is a biomarker for iron burden within the brain, we see that patients with higher levels of ferritin are associated with more rapid metabolic decline, measured on the y-axis here by change in fluorodeoxyglucose PET. So I'm citing down here all of the publications that we had over this period. We drew a conclusion that it looks like high iron levels in Alzheimer's disease would be bad for you and are associated with more rapid cognitive decline. Now, why would iron be bad for you? It's been discussed obviously a bit this morning already. I just wanted to just review the core chemistry of ferritosis, that iron reacts with polyunsaturated fatty acids and it becomes oxidized and generates lipid peroxides. The lipid peroxides have a radical chain reaction which leads to the disruption of the cell and the death of the cell. And it can be blocked by small molecules. Some of them are extremely potent, active in the low nanomolar range. And there's also endogenous molecules like vitamin E, which blocks the lipid peroxidation propagation. Obviously, lowering iron levels have got a chance of blocking ferritosis, at least in vitro. In vivo, there's still some questions about that because you don't need very much iron in order to initiate this reaction. This reaction probably happens all the time in life and is blocked by a system of quenching, which I'll show you in a second. But in terms of pathology, lipid peroxides can generate a lot of nasty chemical reactive species such as 4-hydroxynonenal, malondialdehyde, and these are highly chemically reactive and can do things like protein cross-linking, and there's evidence in the literature that this contributes to the generation of pathologies such as plaques, tangles, and Lewy bodies. But in terms of what stops this from happening all the time in life, iron is buffered by glutathione, which is highly abundant in millimolar concentrations in the cytoplasm, and glutathione is also the substrate for the checkpoint for ferritosis, which is glutathione peroxidase 4. So this is what normally stops this reaction from killing every cell in the body all the time. Now, we became very interested in David de Vos's clinical work on iron elevation and the relationship with Parkinson's disease and his hypothesis that it would be a potential drug target. He had successful phase 2 trials, which I mentioned earlier, which were published in 2014, the FAIRPARK 1 study, which showed that treatment with deferoprone lowered nigral iron levels. This is the area that is dying off in Parkinson's disease. And treatment with the deferoprone improved the outcome over an 18-month period. So a very inspiring clinical trial which got us very excited, and it was replicated by the British group in London, David Dexter, so there was a lot of enthusiasm about the possibility that iron chelation could be used for the treatment of Parkinson's disease. Now this inspired us to try out the same idea for Alzheimer's disease. And so soon after that publication from David came out, my group, I applied for grant funding to do this, the 3D trial, deferoprone to delay dementia. This was partly funded by the Australian government funding system as well as funds from Apopharma, the producer of deferoprone. So it was a double-blind, randomized, multicenter phase 2 clinical trial of patients who are amyloid positive with mild cognitive impairment or mild Alzheimer's disease. And they were treated with deferoprone, 30 milligrams per kilogram per day, or placebo. The dose of deferoprone was the same as David used in his clinical trials, his phase 2 trial for Parkinson's disease. And they were randomized 2-to-1 for deferoprone to placebo. And aiming for 171, we did a power analysis of being able to see a 20% change in cognition over a period of 12 months. And we started in 2018, which was a very fateful date to begin a clinical trial that was going to last possibly several years. The primary endpoint was the neuropsychological test battery with measurements at 0, 6, and 12 months. So this is a composite of various domains of cognition. And the secondary endpoint was QSM for brain iron, volumetric MRI, and safety, of course. And that's where the funding was to do a 5-year study. So what happened? Well, we started recruitment, and this is the consort diagram, and there were a couple of problems. Firstly, of course, we started in 2018, and then there was 2019, and suddenly 2020 came, and COVID caused an enormous problem, because these are elderly patients who are going to a public hospital system, and so it became very hard to get the patients to come to the trial sites, and it slowed things down enormously. And the other really strange thing happened was that the owner of Apopharma was murdered. And as a result of that, anything can happen. If you're going to be a researcher, you have to be prepared. Yep.
He was hanged.
He was hanged, yeah. Yeah.
It
was a home invasion, yeah.
The Apple Pharma owner, okay, who's one of the main sponsors of buildings here in the university, he was hanged by, nobody knows what happened, he and his wife.
Yeah, still.
No, Michael Spiro, the boss of Michael Spiro, the big boss.
Anyway, so unfortunately, therefore, the company was,
Toronto.
yeah, the company who had sponsored us and were giving us the drug was sold off to another company, and the new owners of this company were not interested in continuing the commitment to complete this clinical trial, so they pulled out. So we were left with a perfect storm of a catastrophe, and I thought that we were going to get no valuable information out of the clinical trial, but we persisted, and we managed with whatever resources we had left to actually finish testing 21 placebo patients and 33 deferoprone patients. I thought this would be so underpowered that we would not be able to get a result, but we still went through the whole process and did the analysis anyway. What we had was a very nicely matched cohort of deferoprone and placebo subjects, so we could do some get some valid data. And indeed, there was a lot of valid data, even though the trial was smaller than we intended. And it was just published last month with my colleague Scott Ayton as the first author in JAMA Neurology. This shows you the iron levels in the hippocampus over the 12 months of the study, that as expected, the iron levels rose in the placebo-treated patients, but fell significantly in the deferoprone-treated patients. So it's very nice that we've got target engagement. So here's the primary result, the neuropsychological test battery composite. The placebo group deteriorated slightly over the 12 months, as was expected, but the deferoprone group deteriorated dramatically over the 12 months with a Cohen's d effect size of 0.7. So it's a moderately strong worsening of cognition that occurred with deferoprone treatment. When you break down the neuropsychological test battery into its different domains, we found that the deterioration was not due to worsening memory performance on the 3 different tests that we used. There was a little bit of worsening in attention in the deferoprone-treated group on the Cogstate reaction time, but not the detection component of it. Where most of the deterioration occurred was in executive function. And we see in the 3 subtests that we used, the WAIS, the COAT, the DCEFs, that there was significantly worse performance in the deferoprone-treated group, and which was statistically significant. So the compound was basically making cognition worse by worsening executive function. And this was matched by increased atrophy of the frontal lobe areas, which is consistent with the executive function loss. The insula, the pars triangularis, the medial orbital frontal cortex, the lateral orbital frontal cortex, each had a greater loss in volume over the 12 months of the study. So it was very consistent that the deferoprone was poisoning the frontal lobes. So as we had completed that trial, around about that time, David published his phase 3 clinical trial of deferoprone in Parkinson's disease, and I was very honored to be a collaborator on this trial. It was published in New England Journal in 2022, and David will talk more about it, obviously. Now, he also got very disappointing results, that deferoprone made the composite score of the UPDRS worse over the 40 weeks of the study, and this was attributed to removing iron from tyrosine hydroxylase, which is needed for the synthesis of dopamine, and there was good evidence provided for this mechanism in the paper. So obviously, both David and I shared in the sadness of having these disappointing results, but at the same time, we got very clear information coming out of these major clinical trials. And it's the big question was, at least for Alzheimer's disease, why does correcting elevated brain iron worsen Alzheimer's disease? And so we've done a lot of thinking. We have to recast our whole model about what does elevated brain iron mean, at least in Alzheimer's disease. And one clue about what's going on has to do with the observations, and this is one paper that has reported it, of decreased brain glutathione levels in the tissue in Alzheimer's disease. And this is quite a robust finding. It can be done with MRS, which you can see here in this paper published in 2015. And as you may recall from the ferroptosis reaction, that glutathione is an important component that prevents ferroptosis from occurring. So if glutathione levels fall, and here it's quite a conspicuous fall, which we've corroborated, about 50%, then it's going to make the tissue more vulnerable to ferroptosis. So why does glutathione, I misspelled that, I'm sorry. Why does glutathione fall in Alzheimer's disease? Well, here's the synthesis reaction that glutamate and cysteine use ATP to form an intermediate with another ATP. It then goes, and glycine, it then goes on to make the glutathione molecule. So energy is needed in order to make this reaction, which is ATP dependent. And if you look in the literature, and I just cherry-picked a couple of papers that are out there, if you poison the mitochondria, you get an elevation of iron in the cell and in the mitochondria. And this is an example of iron levels in cells that have been treated with rotenone, a model for Parkinson's disease, by poisoning complex 1 in the mitochondria. You see this dramatic increase in iron that occurred in these cells. It's lowered with a chelator. This type of result can be done genetically, where you make the complex 1 insufficient, and you get, again, an elevation in iron in the cell. There are many instances of this in the literature. And it's been observed, and the significance of it hasn't really been appreciated. But you consider that in these complex 1 inhibited models, that the ATP production must be decreasing, and so what is happening to the glutathione? The glutathione must be lowered. Now, I'm very pleased to have met Slil Ast, who has come to this conference, and I was very impressed with her paper recently in MPJ, Metabolic Health and Disease, which reviews what iron is doing in mitochondria, and makes the point that the iron is sent mainly in 3 directions: for the synthesis of heme, for storage in mitochondrial ferritin, and for the production of iron-sulfur clusters. And the iron-sulfur clusters are essential for the complex 1 and other activities. Some beautiful figures from this paper that's showing the frataxin and the assembly of that produces the iron-sulfur clusters. Iron represents about 40 to 50% of the total iron in the cell is in the mitochondria. So it's a very heavily iron-dependent organelle. And disruptions in mitochondrial iron-sulfur cluster biosynthesis lead to the activation of the iron starvation response, elevating intracellular iron and the susceptibility to ferroptosis. This is a quote from her paper. And this struck me as really being a very good hypothesis to explain why we made things worse by depriving the tissue of iron in Alzheimer's disease. So here's the model of what we're thinking, that in health, this is a very simplistic model, but iron is needed for ATP production by supplying the mitochondria with the iron to produce iron-sulfur clusters, and also heme is produced, and the ATP is used to make glutathione, and this is a normal brain with a normal amount of iron staining in it. And keep in mind that complex 1 needs more than 25 iron-sulfur clusters, so it's very, very hungry for iron in order to create energy. Now, obviously, what we're thinking is that in Alzheimer's disease, maybe the upstream lesion is in the mitochondria. And as a result of mitochondrial failure, you get 2 things happening. Firstly, you get obviously less ATP because the mitochondria are not functioning properly, and because of the drop in ATP, you get a drop in glutathione levels. And at the same time, you get the iron starvation response so that the iron levels go up. Now, to make things worse, this also feeds into the proteinopathy in Alzheimer's disease. In particular, tangles, where it's understood that the highly negatively charged surface of hyperphosphorylated tau attracts iron, which is redox active, and we hypothesize could be a source of radicals that can lead to ferroptosis and the death of the cell. And by this stage, you see there's a lot of iron in the tissue. It's in the plaques, and it's in the interstitial cells, it's in the microglia, it's in the neurons, it's everywhere. So therefore, our model suggests that removing iron could indeed worsen glutathione depletion and increase ferroptosis, because you don't need much iron to induce ferroptosis. You just need catalytic amounts of iron for this to occur. Now, other things that we've noticed in trying to review the literature around these ideas is that mitochondrial iron accumulates with age. It's a nice paper showing the mitochondrial iron levels in rats as they age to the age of 37 weeks. On the lower panel is the calcium levels, which don't change, but it's clear iron accumulates within the mitochondria. And this leads into observations that, a huge number of observations down the literature, that brain iron levels rise with aging. They rise developmentally, and then they continue to rise post adolescence in adulthood, and they get higher and higher throughout the human lifespan. These are a number of papers that have shown that in rodents and in humans, and using imaging of humans. There's a lot of good MRI data now about iron deposition in the brain across the human lifespan. And Peter Nestor is a collaborator of ours who's done a lot of beautiful work on this. So we've always wondered, is this bad for you? We always thought this might be feeding into Alzheimer's disease, but at the same time, maybe it reflects a constitutive degradation of mitochondrial activity with aging. Now, does it generalize? Because iron elevation and glutathione depletion is seen in tandem with neurodegeneration in other diseases, and this is very interesting. So we've published a couple of papers about iron elevation in schizophrenia, both in post-mortem human tissue, and this was with Amit, and also in vivo using 7 Tesla MRI, and this was done with Chris Pantelis in Melbourne. We see this elevation in schizophrenia, and schizophrenia is also characterized by a drop of glutathione in the tissue. In bipolar disorder, there is, and mood disorders, there is a relationship with brain iron. This is one of our papers in molecular psychiatry with Peng Li as the lead author, as the first author, where humans who are at ultra high risk of psychosis in adolescence were treated with lithium, and we used relaxometry to measure the iron levels in the substantia nigra, and showed that over the period of 12 weeks of treatment, the treatment with lithium elevated nigral iron. The decrease in relaxometry signal means an increase in the iron content in that nucleus. And there's a depletion of glutathione in mood disorders, which has been perhaps not as robustly reported as in, say, Parkinson's disease, which I'll talk about next, but it's reported there as well. So again, this tandem increase of iron with decrease of glutathione. And in Parkinson's disease, this is very, very well established. Decreased glutathione, increased iron, is in Parkinson's disease. That's David's work about the imaging of the iron in this nucleus, the substantia nigra. Now, we also have had a play with intervening with, in clinical trials by giving N-acetylcysteine, which we, with Michael Burke, published in 2008, a clinical trial in schizophrenia, 6-month treatment with N-acetylcysteine improved outcomes. This has been since it's been tested out in other clinical trials with mixed results. If there's an effect, it's relatively weak, but we have thought about it. Same with bipolar disorder, that N-acetylcysteine improved depressive symptoms in bipolar disorder. Again, we did that with Michael Burke. And Ray Swanson has led the interest in trying to improve the levels of glutathione in the brain in Parkinson's disease. So there's been interest in these pathways as potential therapeutics. Well, I came across this very interesting paper showing that in neural precursor cells that were studied for mitochondrial respiration using the seahorse system, that treatment with lithium caused an improvement in the oxygen consumption rate, and so lithium improves the metabolism of the mitochondria through a mechanism which is not clear yet. But I just thought I'd mention it since lithium is obviously the major treatment for bipolar disorder, an important psychiatry drug. There's been growing evidence for ferroptosis in schizophrenia. This is a paper which was recently published by a Chinese group which shows that in postmortem human tissue from the prefrontal cortex there's an increase in iron levels. There's an increase in malondialdehyde, which is a lipid peroxidation product, which indicates that ferroptosis is occurring. There's a decrease in glutathione peroxidase 4 activity, which is the checkpoint for ferroptosis, and there's a decrease in glutathione. This was first proposed by Kim Do, but is now quite a robust feature. So we wonder whether schizophrenia has got this same tandem problem of iron elevation with a drop in glutathione, and whether that may contribute to the atrophy that occurs in the brain in chronic schizophrenia. So what can we do about this? Obviously, deferoprone was disappointing for Alzheimer's disease, and there will be discussion, of course, about whether the deferoprone dose was too high. But meanwhile, there are other approaches out there, and one that I'm very interested in is this compound, ATH434, which is manufactured by Alterity, a company which I have shares in, which are not worth very much money, but I have shares in it, so I have a conflict of interest. So it is called an iron targeting compound or a chaperone, and they use these terms very deliberately. This paper was recently published by Dan Kosman's lab, where he characterized the affinity of the molecule for iron species and showed that the KD for ferrous iron is 4.6, and for ferric iron is 0.5. And his argument was that having a micromolar iron 2 binding affinity matched to proteins that drive ferrous iron utilization within the cell indicates this drug could contribute to metal delivery within the cytosol or within the matrix for iron cluster assembly. So this sounded very interesting to me. It happens to have antioxidant properties, and he's saying that it can act as a chaperone for iron shuttling within the mitochondrial matrix. In contrast, he says, to deferoprone, where the KD for ferrous iron is 15 micromolar, so it's not really truly ideal for the labile iron pool concentration, which would be in the order of 2 micromolar. And the KD for iron 3+, has been measured in these reports somewhere between 10 to the minus 25 or 10 to the minus 35, so it's a very, very strong affinity and is capable of removing iron from ferritin stores. So this is a true chelator in that sense, and ATH434 is not capable of removing iron from storage pools. So that's a big contrast. Alterity has tested out the proof of principle of their iron chaperone approach in an indication called multiple systems atrophy, which is one of the Parkinsonian type diseases, which is quite characterized by autosomal dysfunction, cerebellar damage, and extrapyramidal symptoms. And in terms of neuropathology, there's an accumulation of alpha-synuclein as flame-shaped deposits within oligodendrocytes. So very strange but very specific pathology. And David Stamler from Alterity presented data at the 24th recent NDS conference where they presented the iron elevation within the affected nuclei in MSA. And they characterized a rating scale for the severity of MSA, which is called the Unified MSA Rating Scale, and showed that there is an association between the severity of the disease and the amount of iron in the substantia nigra. So they used this rating scale in order to conduct a phase 2 clinical trial, which I'll tell you about in a minute. going back a little bit, my lab played a role in the original studies of ATH-434 when it was called PBT-434, the name of the company changed. And in vitro, if you took alpha-synuclein and incubated, you'll get fibril formation, which is accelerated by the presence of iron, which is the orange, but could be mitigated by co-treatment with 4234. And there was another control, which was 434, which had a methyl group blocking the ligating oxygen, and that was unable to stop the aggregation. This matched the kind of fibril formation that the alpha-synuclein was making. You see the increased fibrils in the presence of iron, abolished by 434, but not abolished by that mutant form of 434. In animal models, this is the MPTP model. There was a dose-dependent rescue of the number of substantia nigra neurons with 434, and a concomitant improvement in the POL test, which is a motor test as a Parkinson's disease model. So all very encouraging. This is a heat map of the iron within the brain in an animal treated with MPTP. You see the iron increased in the substantia nigra and elsewhere, which is rescued by treatment with 434. Levels of 8-isoprostane in these animals were lowered by 434, which is a marker for ferroptosis. And alpha-synuclein levels within the tissue were decreased by 434. Indeed, giving dogs 434 for 28 days lowered CSF alpha-synuclein levels, and rats given a single treatment of 434 lowered alpha-synuclein levels. So it was a very interesting drug in many ways. A more recent paper replicates this aggregation result with showing that 434 can block the iron-induced aggregation of alpha-synuclein in cell-free models. But more importantly, there was a mouse model where human alpha-synuclein is overexpressed in oligodendrocytes. It forms this pathology, and it causes substantia nigra death. And 16-month old animals who have been treated and untreated with 434, you see the drop in the number of neurons, which is rescued by treatment with 434 at different doses. And the iron levels, which are elevated in the model, are also remedied by the highest dose of the 434, which is tested. So all that looks really nice. Dan Kosman presented at Society for Neuroscience in 2024. All of these data are in the public domain and are found on the Alterity website. It's not published in peer-reviewed journals yet. He showed that 434 could block ferroptosis by, induced by menandione, where the BODIPI, which is a measurement of lipid peroxidation, was suppressed by 434 as it was by the reference compounds Trolox and tocopherol, which are vitamin B analogs. And he also showed seahorse data from mitochondria, which I found very interesting, showing that 434 could improve the respiratory activity of mitochondria. So that was very encouraging to see that 434 could potentially indeed be acting as an iron chaperone to increase iron sulfur cluster synthesis, which is the big question there. Is this result explained by improved iron sulfur cluster formation? There's no data about that yet. So finally, this is where I'm going to end off. The company announced the results of a phase 2 clinical trial, which has not been published in a peer-reviewed journal, but it's on their site, you can see it. A randomized, double-blind, placebo-controlled trial of early-stage MSA, 12-month treatment with 434. 77 adults were treated at 2 different doses against the placebo, 26 sites in 6 countries. And the results are that it produced a clinically and statistically significant improvement on the modified rating scale that they used, a 48% slowing of clinical progression at the 50 milligram dose, which was significant, a trend towards improvement at the higher dose. And the higher dose showed us 62% slowing of progression at week 26, and the 50 milligram dose improved clinical global impression of severity significantly. They showed a reduced accumulation of iron at the 50 milligram dose in the putamen by MRI, and they've got other biomarkers that are going to be reported. the safety profile was very favorable. So, in summary, the model that we're contemplating now in Melbourne, and the one that we're testing out in the laboratory, is that this is the healthy iron pathway. Iron is needed for ATP production, which is needed for glutathione production, because iron is needed to make iron sulfur clusters in the mitochondria. But something goes wrong in these diseases where an iron starvation response occurs. The mitochondria can't make enough energy, the glutathione levels fall, and that enables ferroptosis to occur. At the same time, the elevated labile iron pool is available for interacting inappropriately with other proteins within the cytoplasm, and this either forms the pathologies of tangles, Lewy bodies, SOD1 accumulations, TDP43 accumulations, lipofuscin, and maybe can feed into inappropriate contact with lipids and ferroptosis. But the main message is that removing iron could worsen glutathione depletion and increase ferroptosis. So we need to be very careful about how we approach chelation in neurodegenerative disorders. You want to move the iron, not remove it. Okay, so that's the acknowledgement. The team in Melbourne and all of the people involved in the clinical trial and our collaborators around the world. I'm delighted to be collaborating with Amit and his team on schizophrenia now. Of course, with David, we've been friends and colleagues for so many years, and Julie Schneider's team in Chicago and others around the world that we've had very happy collaborations with. So thank you very much for your patience.